in today's kind of environment, we understand things like measuring antibodies, but you have to think over 130 years or 120 years ago, sorry, that the understanding of what was in our blood, what was in the serum, wasn't very clear. And so it wasn't till some biochemists ultimately figured out that in our blood there are these proteins. And so you can see that I've kind of broken down two of the early Nobel Prizes were for this description of that we could essentially vaccinate animals and that would lead to some sort of protective property in our blood. But then if we fast forward, 60, 70 years here, people started to realize that this protective property was actually due to proteins within our blood. And so in the early 70s, the actual structure of what an antibody is, and that's what's shown here on the bottom left, that this Y-shaped molecule, which is always kind of detected as a Y shape, and we'll talk about why that is a little bit later, is what we now think of as antibody. And that if you jump ahead to the 80s, people started to learn about how to produce antibodies in the lab. And this was one of the big stepping stones towards making antibodies as therapeutics. And then in the late 80s, people actually discovered how our body made antibodies. And we'll talk about some of the details here. Okay, so many people learn about antibodies in the first place because this is central to what we call our adaptive immune response. And what I mean by adaptive immune response is that many of you guys learn early on that if we get sick with something and we recover from that, in theory, we won't get sick from that same thing again. And that's, again, the basis behind vaccines. So we call this phenomena immunologic memory. And that means essentially our immune system remembers what we've seen in the past so that if we encounter this thing again, we can basically clear this thing much, much more quickly than the first time we saw it. So if we break it down, the first time we see a pathogen or a vaccine in kind of modern medicine, we develop a response, but it takes a few days. Right? And during those few days, sometimes the pathogen can proliferate and make us sick. But ultimately, we will clear it. And what we leave behind are what are called memory cells. And so if you look at the figure in the bottom right picture, you can follow the blue. If we get exposed to a pathogen, just called antigen A here, we're going to develop an immune response. But that immune response does take a few
how are you?
in order to develop.
Okay.
Right? And once we develop that immune response, it will come back down because we've cleared that pathogen. But you can see it never goes down to where we started. It leaves behind a population of cells, and these are what we call memory cells. And that these memory cells can continue to make things like antibodies. Now, the trick is if we see that same thing again or something incredibly similar, it doesn't take nearly as long for our immune system to get going again. And I think screen sharing just, did the Zoom die? Okay, so I need to screen share again. Okay, we're back. So the point is if we see that same thing again or something very, very similar to it, you can see that the time it takes to develop that immune response is much shorter and the response is much, much more dramatic. So this is the hallmark of what we call an adaptive immune response. It has a memory, and this is why vaccines work, right? The vaccines are to mimic the first time we see that thing. It takes a longer time to develop, but it leaves behind that memory. And that when we look at what the memory leaves behind in the news, again, much of what we hear about are antibodies. And so we can literally measure the abundance of antibodies that are left behind from either that first exposure or a vaccine. And you can see that a subsequent exposure to the same thing or something similar gives you a much higher and much faster response. Okay, so this is what an antibody looks like structurally. Like I showed you before, it's these Y-shaped molecules. They're actually made up of 4 individual proteins. Okay, sorry about that. So we okay, so there are these Y-shaped molecules, and that the antibodies within us, right, have been developed based on our lifetime exposure to all the different antigens that we've seen. Some of us have seen more than others, but in general, if we look at our antibody profile, they will reflect all the different things that we've encountered in our life. And if we look at all these different antibodies, structurally they all look like this Y-shaped thing, but one of the major differences are the very, very ends or the very tips of the Ys. You can see the little V, that V stands for variable region. So all of our antibodies in us have this kind of stereotypical Y shape, and that they bind to different things because they have different V regions among them. Okay, so that variable region is why different antibodies can actually bind to different things. And what I was talking about before is that our adaptive immune system learns based on what we've been exposed to. And that's why we all have slightly different resistances to different things, because again, we've all had a different history of what our immune system has seen. So this implies that antibodies themselves aren't already encoded in our genome, ready to go the moment we're born. Our antibodies are actually created slightly differently by all of us, and we'll talk about that process in a little bit. So the easiest way for me to kind of frame the idea of how antibodies are made is that every B cell, so a B cell is the cell of your immune system that makes antibodies, it independently basically cuts and pastes the DNA inside of it to create a novel specificity. And we'll get into a little bit more details there. But what this picture is showing is that we start with a B cell precursor, so this is found in the bone marrow, and that as this cell develops, every individual cell will randomly make some sort of choice to create a specificity. Now, there's no knowing if that specificity is going to be useful or not. This is all a random process. But this is happening in millions and millions of individual cells all the time. Since the specificities are random, the first thing you actually want to do is make sure that that specificity doesn't accidentally recognize self, right? And recognizing self would be bad. And that's actually the foundation of why some people develop things like autoimmune diseases, because they have some problem where their immune system has lost the ability to distinguish self versus non-self. So in general, during this process, we get rid of cells that recognize self because we don't want to lead to autoimmune diseases. And that means that what's left are what we call our mature B cell population. So these are sent out into the body, and they're just going to circulate around. Now, what happens is that if we get exposed to a pathogen, there are only a few cells that will actually respond to that pathogen. But when that cell responds to that pathogen, it will proliferate and basically make more copies of itself. So what's functionally happening is that we create what's called a repertoire, which is this huge array of B cells that recognize all sorts of different things that's done randomly. We get rid of ones that recognize self, and then the ones that are left are the ones that circulate around, and these are our naive cells. If they ever encounter a pathogen that they can recognize, only the cells that recognize the pathogen will actually expand and then start making antibodies. Okay, so what that means is that when we get vaccinated, it's not that we create the specificity, it's simply that we activate the cells that were already there to begin with, and they will proliferate and make more copies of themselves. Okay, so how does this all happen? So if we look at the DNA inside every single B cell, they come with these little cassettes. So these are what are called the V regions, D regions, and J regions. And there are about 40 V regions, 25 or so D regions, and 6 J regions. Now, if I asked everyone on this Zoom, to pick a number between 1 through 40, and then I asked you guys again to pick a number between 1 through 25, and then I asked a number 1 through 6, it's pretty unlikely that anyone would pick the same 3 numbers, right? And so that's kind of the basis of how B cells create random specificities. Each cell independently chooses one V, one D, and one J, and the DNA is literally cut and pasted together to make that antibody. Now, this happens on both what's called the heavy chain and the light chain of an antibody. So simply by choosing a different V, a different D, a different J, and each B cell doing this independently, you can see that you can come up with a lot of different combinations of things that these antibodies can actually bind. And that's what we call combinatorial diversity. It's basically a numbers game, right? You just choose one of these, one of these, one of these, throw them together, and that creates a unique specificity. And that's where that randomness comes into play. Now, there's even more randomness in the system in that what happens is when those V's, D's, and J's are pasted together, there are special enzymes in the cells that start adding random pieces of DNA, right? And that creates even more randomness. And so when you start factoring all the possible combinations and the fact that you start throwing in random, what are called nucleotides or pieces of DNA here, you can get a huge amount of different types of antibodies by doing this, right? So you're probably more limited by the number of B cells than you have than the possible combinations of all the different things that you can create. And that this is, again, our repertoire. This is what we have even before we get exposed to anything. And then when we do get exposed to something, only the cells that can recognize that pathogen will activate, proliferate, and then create antibodies. Okay, so understanding how these antibodies work, basically, we know now that we can take antibodies from one person, right, and that we can use them in other people, right? So many of you guys might know a few examples of this already, right? If you've ever been bitten by a snake, right, you can go to the hospital and they will actually give you antibodies that can bind and neutralize the toxin that snakes give you when they bite you. So there are a lot of examples. This has been done for several decades at this point. But it's becoming much more common simply because we're much better at making antibodies now. The benefits of these is that you can have an instant effect. You don't have to wait for your immune response to make your own antibodies. You can just passively give antibodies either created in a lab or from someone else. Right? And then there are people born out there with genetic mutations that might have weakened immune systems. And because of that, they can't actually make good antibodies. So you can actually just transfer antibodies to them. Now, the big downside about transferring antibodies is they don't last very long, right? If you put antibodies into someone, yeah, they can last for maybe a month or a month and a half, but after a while, you're going to have to keep giving them antibodies. So they're not really a replacement for a normal memory response that you would develop through an exposure, but it's not bad for a short-term protection. This really came to kind of prevalence. There have been Ebola outbreaks where one of the ways that people realized that you could protect people short-term was if somebody got infected with Ebola and they happened to survive and clear it, you could take serum from those people, therefore the antibodies from those people that survived, and you could transfer that serum to at-risk people, healthcare workers, and that could protect them in the short term. So again, the idea of transferring what we call convalescent serum from people that have survived and cleared various pathogens can work, and it does work in practice. The downside is there's a finite supply, right? It depends on having people available that can donate the serum, and that there is variability, right? One person that survives Ebola will have antibodies, but those antibodies from that one person aren't going to necessarily be the same as somebody else who also survives. So there's variability depending on who you get it from. Okay, so one of the Nobel Prizes that I talked about earlier was learning how to make antibodies in the lab. And so this was done in the 70s. in that they can isolate individual B cells that make the antibodies, and then they can grow those B cells in the lab indefinitely. And that means that we can basically, for example, immunize a mouse, find the B cells that make antibodies what we care about, and then one of the things we have to do before we start putting it into humans is we have to make the mouse antibody look more like a human antibody. Because if you put a mouse antibody into a human, the human's immune system is going to recognize the mouse antibodies as something foreign and mount its own immune response. So typically that mouse antibody has to be converted into more human-like antibody. So one level is making it chimeric. Remember, the tips are the variable regions. So that's the only part that really matters. You can do it even more specialized way, the part that actually binds, or now there are some companies out there can actually make fully humanized antibodies in the lab. So ultimately, the ability to make antibodies in the lab, these are what are called monoclonal antibodies, meaning that we know exactly what the specificity is, what it actually binds to, what the sequence is. These are what are currently being used in a lot of the current therapies out there. So as an example, with the coronavirus pandemic, one of the things you may have heard of is a company called Regeneron, and one of the therapies that they designed was this REGEN-COV, and what this is actually antibodies, right? So normally in an infection for coronavirus, we have a cell and we have the virus, and the virus physically interacts with the cell to infect. And that this company started to create antibodies that would bind to the virus, and that would essentially prevent infection. So the beauty of this idea is that you could grow these antibodies in huge amounts in the lab, and that these antibodies are specific to the coronavirus, and that you can just inject the antibodies into people. This would result in instant protection from the virus. If you were already infected, it would, in theory, prevent the virus from continually infecting new cells. But the downside is this sort of therapy doesn't necessarily last very long, right? You inject antibodies into someone, but they're not going to stick around forever. And the other problem is you spend a lot of time engineering these antibodies to recognize this virus. If this virus happens to mutate so much that the antibodies don't recognize it anymore, then that therapy is not going to be as effective. So these sorts of therapies have been useful for certain subsets of people, people that are immunocompromised, or people that are really sick and need some other therapy. Their own antibody production wasn't enough for whatever reason, that these basically exogenous or extra antibodies that were injected into them can really help them clear these sorts of viral infections. So I think this is a good example of how some of these antibody therapies can be used to prevent infection. I think vaccine approaches are obviously typically better, more efficient, but the downside is a vaccine typically will take time to develop enough protection. In this system, you literally have antibodies in a bottle that you can inject into someone, and they are effective right away. And so I think this has hit the mainstream, but I think many people don't realize that antibodies have been in use for a long time. Here's an example of a drug. This is what's called Humira. It's classically used for rheumatoid arthritis. There are a few other diseases that Humira has been used for. And so this is the idea here that, for example, like rheumatoid arthritis is a chronic inflammatory state. Here we have something called a TNF receptor. Don't worry too much about the details, but what happens is we have a molecule called TNF, and it binds to the TNF receptor, and this is actually what leads to inflammation. So several decades ago, a company thought, well, if we could develop an antibody that binds to TNF alpha, then if that antibody binds to TNF alpha and it prevents the ability of the TNF alpha to actually bind to receptor, then you would block the inflammation, right? So Humira was one of the antibodies that was ultimately developed to bind to TNF, and this is, again, a FDA-approved therapy to combat things like rheumatoid arthritis. And so here, I wanted to show a little bit of data. I don't always show data. This is obviously not my data, but this is what some of these clinical trials look like. So normally when a drug like this comes to market, what they want to do is, does this thing have an effect over a placebo, right? In this case, the placebo is given patients are randomized. And in this particular example, what they were trying to do is they were trying to test different concentrations of the antibody and also how frequently the antibody would be delivered, right? This is classically how clinical trials are done. They will try different concentrations just to see what works the best. And typically in this sort of situation, none of the patients knows what they are getting. They don't necessarily know if they're getting the low dose, the high dose. They might be getting placebo. And so what you then do in this case for rheumatoid arthritis is they will get these treatments, and then they're going to ask the patients, how do you feel, right? They will do more objective measurements about joints and joint swellings and things like that. But what we can start seeing pretty quickly is that after they collect the data, they will go back and assign the different patients these different groups, right? And you'll see pretty quickly that most of the people on placebo, yes, a few of them started to feel a little better, right? But you can see that the group that started feeling the best were the ones that got the higher dose more frequently, right? And so these are blinded clinical trial studies that really demonstrate that these antibodies can have a pretty profound effect on, in this case, rheumatoid arthritis. And the next example here is a similar idea where there are antibodies that are currently being used to do what's called immunomodulatory therapy or immunotherapy. And these are antibodies that are designed to try to help the immune system combat cancer more effectively. So if you've ever heard of a drug called Yervoy, this is actually an antibody. Opdivo is a different antibody, but it's the same idea where this is looking at metastatic melanoma. So the survival prognosis for metastatic melanoma is not particularly good. And so you can see this is overall survival over time. And so if we start here, everybody starts at 100% when the clinical trial starts, and then they measure survival. And what you can do is here is essentially a control group, and you can see that the people that get the antibody therapy, they live a little bit longer, right? Obviously, many are still dying, and melanoma is still one of the real challenges for oncologists. But you can see it does make a measurable difference. And then this next study is showing if we add this and another antibody, so now we're adding two different antibodies, you can see that that curve gets shifted up even higher, showing that survival in general is much better with these sorts of antibody therapies. Now, again, these antibody therapies have really become popular in, I would say, the last 10 years. They've really hit the market. And so here's kind of an example. So these are FDA-approved antibody therapies. Many of them are for cancer or cancer-related diseases, and that's what's shown here in the red. But within the last 10 or so years, you can see here the bulk of the antibodies receiving FDA approval. I just put a couple down here. These are probably the more common ones, if you watch like, you know, the evening news, these are what you hear a lot of the commercials for. But if you look at, there's always a brand name for a drug, and then this is what the non-proprietary name, or usually when they undergo clinical trials is what they called. And one of the things you notice is the last three letters here is MAB. MAB basically means this is a monoclonal antibody, right? And so all of these drugs, many of them you guys might have heard of, classic ones. Humira is the one I talked about. Herceptin is one that's used for breast cancer. Rituxan's used for non-Hodgkin's. And so these are all antibodies that are now FDA approved, and that I would say this is one of the biggest growing sectors in the biotech industry, simply because antibodies, we know they're safe when we put them into people because we already all have antibodies. And the key is, can we develop good antibodies that can bind to things that can basically help our immune system perform more effectively? Okay, so sorry about all the kind of the technical difficulties. Hopefully I didn't go through that too quickly, but I'm happy to take questions with you. This is just kind of a summary of what I've talked about, is that, again, individual B cells will make antibodies, and this is this process, what we call VDJ recombination, and that only the cells that recognize the antigen are actually activated, and these are the ones that are actually making the antibodies. We have memory cells, and this is why antibodies stick around for a while, because they're going to continually make antibodies. We can passively transfer antibodies, and that can protect us from different pathogens. And ultimately, again, these antibodies are being heavily researched now because they can be really useful as treatments against autoimmune diseases and especially cancer. Okay, so with that, again, I'm happy to take any questions. I don't know if you just type them into the chat box or what. Okay, yeah, so if you're on Zoom, go ahead and type questions into the chat box. Yeah, so one of the things that people started to realize probably about 20, 30 years ago is that we knew that the immune system played an important role in prevention of cancer, but it wasn't really clear how that was happening. And so what people have learned more recently is that our immune system plays a really important role in prevention of cancer. But once you get cancer, or at least once it's diagnosed in you, your immune system is still trying really hard to kill cancer cells. The problem is when this becomes more of a chronic phenomena, many times cancer cells can evolve to develop um, basically evasion mechanisms or a way to hide from the immune system. And so many of these antibodies are actually designed to try to help the immune system focus in on the cancer cells a little bit more. And one of the other things we notice is that the immune system also kind of gets what they call exhausted, or we call it T cell exhaustion, and that some of these antibodies are trying to prevent the T cells from getting exhausted. And so it's the same idea where these antibodies basically block the signals that would normally tell a T cell to slow down and stop. Because historically, you're trying to find this balance where you have to keep in mind cancer is a self phenomenon, right? We're great at fighting viruses and bacteria because they don't look anything like us, but cancer is us, right? So it's a fine line between our immune system trying to kill off cancer cells, but at the same time not developing some sort of autoimmune type disease, right? And that's that fine line that we typically walk in cancer biology there. Yeah, recombinant is kind of a generalized term that simply means that it's a purposefully made thing in the lab. Historically, so for example, something like a flu vaccine. What the flu vaccine typically is a flu virus that is then propagated in chicken eggs. And that's why they always ask you if you have an egg allergy when you take a flu vaccine. But they grow up large amounts of that virus. And that, at least for the flu virus, they kill it. They either heat it or they treat it with some sort of chemical that basically makes it inactive, so it cannot replicate at all. There are recombinant flu vaccines. If you've ever had an inhaled flu vaccine, those flu vaccines are actually still replication competent, but they've been weakened to the point where they don't make you sick, right? And so those are examples like we can purposefully kind of mess around with the genetics of a virus to make them a recombinant virus, and that, in theory, is a more controlled way to produce them. Hand up.
Hi, can you hear me?
Yeah, I can hear you.
Good. I have a couple of questions. One is the antibody treatment available generally for all these cancers? And second, at what stage of the cancer are these antibodies more effective?
Yeah, so the antibody therapies really depend on what kind of cancer. Some of them have been approved for things like melanoma, A few have been approved for breast cancer and things like that. So it really depends on which type of cancer you're talking about. But I will say that many of them are used in combination with other things. They're typically not used as standalone treatments. And so a lot will, they are readily available in terms of if your oncologist should have access to them if they make sense for you. But it just kind of, it depends on your specific situation. Usually when it comes down to cancer, the strategies are based on a lot of statistics. They'll say that, okay, historically, these sorts of things have worked really well, and that when antibodies or any drug goes through clinical trials for that matter, what they're trying to do is that any better than the current standard of care, right? And that's where a lot of these clinical trials will try different variations to see, does that actually result in more favorable outcomes? But any of the antibodies that have FDA approved, in theory, would be available to anyone if their physician felt like it made sense for them.
And at what stage of the cancer they are more effective?
So again, it really depends on which cancers you're talking about. Since these are systemic therapies, you're injecting them and that they will circulate around your entire body. They could, in theory, be used at any point, depending on it, where you were in your progression. So, for example, I think many of you guys remember when the former president Jimmy Carter, everybody thought he was going to pass away. He was kind of on the last, his prognosis was pretty bad. I think he took one of these antibody therapies and he basically is recovered. And so it really depends on the type of cancer, the situation specifically. Not everyone responds to these antibodies, as you can see in those clinical trials. It didn't rescue everyone, but it definitely shifted the curve so that more people had a better prognosis.
Okay.
Typically, once it's clear that there have been enough cases of something, it's trying to figure out what that standard of care is. So obviously, if you're at a research hospital, you might have experimental drugs available that, a hospital out in a rural community wouldn't have access to. But once these things get kind of full approval, there are typically kind of standard treatments that people would go. Obviously, when you get cancer, not everyone's cancer is the same. So that's why not everyone always responds. So in a lot of ways, it's try what's the most commonly effective treatments. And if they don't work, then try some of the other ones, because it's very, very hard to predict in many cases what will work, what won't work. I would say maybe 20 years ago, when DNA sequencing and the human genome became more common, everyone said, oh, that was going to lead to personalized medicine so that we could create all of these amazing therapies on an individual basis. The reality is that hasn't really panned out the way we thought it would 20 years ago. The information is still important, but I would say there's still a lot of try what the standard of care is. If it works, great. If it doesn't, try something else. Yeah, so every clinical trial in the US has to be approved and that it's entered into a database. And it should be, I think, clinicaltrialsgov. It's a government run database. And you can actually search for a disease, search for a drug, and it will tell you what if there are any, and then where they're at. And it will typically also sometimes tell you the criteria which you need to satisfy to enter those clinical trials. So all of that should be available to the public. I don't think you can just walk in. You might have to get referred to something like that, but all of that information is there and you can search it. Yeah. Cost is a big barrier. I think one of the challenges is that when you start dealing with therapeutics in general, antibodies are classified as a biologic because they are a protein, right? Many drugs that people take in pill form right? They're small chemical molecules. So the production of small chemical molecules is typically much, much cheaper than biologics. Biologics have to be made by living cells. It's not a chemical synthesis process. It's basically taking cells, growing them in really, really large amounts, and then purifying the protein. So cost is a huge barrier for some of these things. Obviously, the cost of a drug isn't just about the production of the drug. It's about the cost of the research it took to get to that point. But that's always been one of the major concerns with the trend towards biologics. These drugs are not cheap. And you and I talked a little bit about things like CAR T cells. Those sorts of therapies are ridiculously expensive simply because those require a lot of basically personalized things. So for those of you guys who don't know about CAR T cells, the idea here is you actually pull cells out of a patient, you genetically modify these cells, and then you put those cells back into the patient in order to try to kill cancer cells. And it's shown some really good promise, but again, it's pretty labor intensive. You need specialized, obviously, techniques and instrumentation to do those things. And that's why those sorts of things are so prohibitively expensive. Oh, you're welcome. Thank you. Oh, my name is Joseph Lin.